In this video, we will be going over the structure of DNA. In 1953, Watson and Crick discovered the three-dimensional structure of DNA by figuring out how the nucleotides fit together. Using X-ray diffraction images and all the available knowledge they had at the time on DNA chemistry, they constructed a molecular model to find out if the chemical principles fitted with the DNA structure. As they were making the model, they found that base pairing was occurring, where adenine could bond with thymine and guanine could bond with cytosine. Once a model was developed, Watson and Crick discovered that DNA consisted of two strands of nucleotides wounding around each other, forming a double helix. The strands were running in opposite directions, meaning that they are antiparallel, and that the double-stranded DNA structure formed in the right-handed direction, with a hydrophilic backbone of sugars on the phosphates on the outside and the hydrophobic bases on the inside. There are many features that are included in the structure of the Watson-Crick DNA. We know that the Watson-Crick model is B-DNA, consisting of a right-handed helix with approximately 10 bases per turn. There are major and minor grooves within the helix, which are created from the spiraling of the nucleotide strands and allow for the binding of proteins that regulate the expression of genetic information. Major grooves are used when the distance of phosphate and sugar backbone between two nucleotides are far apart, and the minor grooves are used when they are close together. Before Watson and Crick's discoveries, it was already known that DNA consisted of nucleotides containing a sugar, a nitrogenous base, and a phosphate group. The sugars have carbon atoms numbered from 1', 2', 3', and 5'. At the 1' carbon of the sugar, the carbon attaches to the base, forming a covalent bond. At the 2' carbon, we can determine whether the nucleotide structure is DNA or RNA. If an OH group attaches at the 2' carbon, then the nucleotide is an RNA sugar, called ribose. If an H group attaches at the 2' carbon, then the nucleotide is a DNA sugar, called deoxyribose. At the 3' carbon, an OH group attaches at one nucleotide and connects to the 5' carbon with phosphate at another nucleotide. Since the two nucleotide strands are antiparallel, the 5' end of one strand is opposite to the 3' end of the other strand. This binding is occurring due to phosphodiester bonds. There are two different types of the nitrogenous base of a nucleotide, a purine and a pyrimidine. A purine has two rings in its structure, whereas the pyrimidine has one ring. There are two purines, adenine and guanine, and three pyrimidines, cytosine, thymine, and uracil. Thymine is found only in DNA and uracil is found only in RNA. We know that the pairing of bases on opposite strands of DNA are possible due to the linkage of hydrogen bonds. However, the main force in stabilizing the double helix of the two nucleotide strands is done by a hydrophobic interaction, which is maximized by base stacking. Let's move on and talk about denaturation. Denaturation of DNA occurs when hydrogen bonds break, causing two strands to separate due to heat or chemical factors. The midpoint at which DNA denatures from temperature is called the melting temperature, or Tm. Now let's say that the DNA molecules are placed in a high concentration of salt solution, such as saline sodium citrate, or SSC. We know that the destabilizing forces in double-stranded DNA are the negative charges present in the phosphate backbone, and that salt ions partially neutralizes these negative charges. The presence of SSC in the double-stranded DNA will decrease the amount of destabilizing force. Therefore, it will keep the DNA strands and its properties stronger together. Because the DNA strands will be more strongly together, it would take a higher level of temperature for the DNA strand to denature. Therefore, it will increase the Tm of the DNA. This is the end of the presentation. Thank you for watching.